As we begin the concept of molecular genetics, we have to start by looking at the structure of DNA and a little bit of background before we can talk about the other bits. And so we start looking at DNA and we think, okay, well, what is it? And it's our genetic information. It's also found in almost all life, and when we think about it, we think of a strand of DNA, which when you find it within it, you have what are called genes. And these genes code for protein. So basically, when you're looking at DNA, you are looking at just the codes for life. It's coding for life. And human genes, or human DNA, for the normal genotype of a somatic cell, there are 46 strands, okay, that represent roughly 3.1 billion base pairs. And what we mean by base pairs is, when we've talked about it at grade 11, quite often you would see that A is linked with T and C is linked with G. Well, those two represent two base pairs. The diagram on the left is a generic looking of one of the base pairs. So if you look and you were to point out things, I'm circling in yellow 1/2 of a base pair. And so when you look at the A on this side, right here, what you're getting is this base pair, which is made up of those 3 points. If you remember what the structure of a nucleic acid has, it has a phosphate group, it has a 5-carbon sugar, and it has a nitrogenous base. All 3 are there, and so if you wanted to sketch it out on the bottom, there's your 5-carbon sugar, and then you have your nitrogenous base here, and then off the 5th carbon, you have your phosphate group. And again, those are the 3 parts to a nucleotide. So that's one nucleotide. And when you join them together on the other side with its complement, it's a base pair. And the other really cool thing, like when you think about that, 3.1 billion base pairs. 3.1 billion base pairs. So in those strands, which we call our chromosomes or chromatid, chromatin. So when you look at chromatin, it stretches out very long, and remember we have 46 strands. If you were to link them and stretch them out for one cell, you'd be looking at about 1.6 meters stretched out for one cell, okay? And that 1.6 meters, again, has those 3.1 billion base pairs. And so when you think about it, I'm stretching this out. It makes that sort of ladder shape. It would be coiled, but I can't draw a nice helix that DNA does naturally. But if I was to zoom in on this area and make that a zoom, I would have a sugar. Oops, I'm having trouble drawing. I apologize. I would be able to make that into something that looks like this. And let's say the one side is A, and then you'd have a phosphate group on that side. This side, there's hydrogen bonds between them. and you would get a T over here. But what it looks like is upside down with the phosphate. Oops, I'm going to do that the right side up so that it stays on the right side. Let's just draw this properly. An O and a P. Okay, now when you look here, you have an OH and an H. And up here, you would have an OH and an H. Okay, and so the free end, this end would be what's deemed as the 5' end, and this end would be the 3' end. On this side, you'd have the 3' end at the top, and a 5' end down at the bottom. And let's take a look at a much nicer diagram, and there it is right there. And we're just going to zoom into this. So when you take a look, what's key up here is there's a 3' end, and there's a 5' end. And you might be asking yourself, what do you mean by a 3' and a 5'? Well, it's talking about the carbons found on the ribose or the deoxyribose sugar. So this is carbon 1, this is carbon 2, this is carbon 3, carbon 4, and carbon 5 is where you get the phosphate attached. Okay? And this has an H, and hence why it's deoxyribose, because it's missing an OH. And this is an OH. If you look at the diagram over there, right here, the free end is the 3. So you can see that the free end, I'm going to highlight it, is the 3' end, whereas over here, the free end is the 5'. And so what happens is it's called anti-parallel when you start looking at DNA structures, because one end will be 5' to 3', and then the other complementary strand will be 3' to 5'. And again, that latter are the hydrogen bonds between the nitrogenous bases. So you'll have A on one side, and then C to G on the other. So what you would get, let's say this was 5', this is 3', and then this end would be 5', and this would be 3'. So if you want to see a full genome, and you think that this, like, right here you would have, like, here's a base pair. And remember that the human genome has 3.1 billion base pairs. Okay? And chromosome 1 has over 200 million base pairs. So when you consider the human genome and how much work time it took for them to code all of the human genome, the 3.1 billion base pairs, with the supercomputers at the time, they did a phenomenal job. So just to recap, remember, DNA is found in eukaryotes in the nucleus. Okay. It runs anti-parallel. It also has a 5' to 3', one of the half of the base pair, and it runs 3' to 5' on the other one. It's a helix, it's helical, so it actually coils on itself. And if it's unwound, remember from grade 11, it's chromatin. If it's wound up into part of a chromosome where it's really, really tightly wound, it's a chromatid. And if two of the identical ones are wound up together and held together with the middle part, that is a chromosome. But for the DNA to be really operational and to find those genes that are in there, like along here and here and here, you need to have it unwound. Genes aren't working when you're wound up into a chromosome. And the last part is, it's roughly 4% of our genome. That total 3.1 billion base pairs, about 4% codes for proteins, for known proteins. So in other words, there's a lot of DNA in our bodies that we're not sure what it does. And that is the lesson on structure of DNA.